Okay, everyone, it's time to start our webinar now. Okay, and good afternoon, everyone. Welcome to today's International Perspective webinar series. And today we are honored to invite an expert from the Temasek Polytechnic, Singapore. And the topic of the expert bring today is the detecting the invisible, the role of the molecular diagnostics during the pandemic. Before the webinar today, I would like to give a brief introduction of our project. The International Perspective webinar series is a series of the international online knowledge lectures jointly led by the CMUTED, China ASEAN Technical Education Collaboration, CATCP, and co-hosted by International Youth Culture and Education, KNG, and GoStudy. This program is expected to invite 1 to 2 school faculty member or the industry expert to give the webinar each month. And the purpose is enhance and reach the growth of the knowledge and broaden the horizons of learners and the scholar in Southeast Asia region and other region by the inviting expert from the different country and different field to provide high quality and universally online lecture that help the learner to acquire the specialist knowledge through the knowledge sharing and cross-culture understanding. So today we are delighted to have a Dr. Ambraj Malados. He is the senior scientist of the Veterinary Science School of the Applied Science in Temasek Polytechnic in Singapore, and is also the research science in Singapore MIT Alliance for the Research and Technology. And his research field is including the CRISPR diagnostic and POCT device, food safety testing, and molecular and sinus biologics, and so on. And today we will have a Dr. Ambraj to bring the a very interesting and meaningful webinar. And after they finish the sharing, they will have a Q&A section. In the Q&A section, you can feel free to ask any question or any points you want to discussing with our expert today. Okay, so now let's warmly welcome the Dr. Ray to give today's webinar. So now you can share your screen. And dear participant, we have sent a virtual background in the chat box. Please try to use this one. Thank you.
Good afternoon, everyone. Are you guys able to see my slide?
Yes. It's okay.
Yeah, thank you. Thank you very much. First of all, I would like to thank Ms. Luna and the organizers of the event for this opportunity. I am glad to talk to people from almost whole of Southeast Asia at any point of time. So this is a very general topic. So feel free to, I know this group is heterogeneous, meaning you have a different backgrounds, not only different subject, but also from different level of studies. At any point of time, if you have question, you can just unmute the mic and feel free to ask question. This is going to be like a nearly 60 minutes discussion lecture. So I don't want to be like monotonous. So I'll be also asking some questions. It is okay to give the wrong answer. Okay, but I want you to just unmute the mic and answer me. Thank you very much. So the topic for my today's talk will be detecting the invisible. You know that what are invisible? These are microorganisms which are not cannot be seen by the naked eye. You need to use a microscope. That is why I use the term invisible. And what is the role of molecular diagnostic, especially during the pandemic? So what you see here is my institution, the main building, and yeah. We are from Temasek Polytechnic, which is one of the 5 polytechnics in Singapore under the Ministry of Education, Government of Singapore. We have about 1,500 staff and nearly 14,000 full-time students and another part-time students. We also offer more than 40 different courses. I belong to School of Applied Science, where you can see here, specifically, I belong to Center for Aquaculture and Veterinary Science. Together with this, we also have Center for Applied Nutrition Services and we have a Center for Urban Sustainability and the Center for Research Opportunities in Plant Science, and we all work together. Moving on. The first thing that I want to discuss is, so what is diagnostic? What is a diagnostic? The term indicates. As you can see from here, diagnostic means detecting, identifying, and monitoring the disease or health conditions using biological or chemical and technological tools. You know what are biological tools? Say, for example, doing some assay, like enzymatic assay, or bacterial culture, or chemical. You can test the blood chemistry, right? Or urine, how much sodium or potassium or other minerals you have. Or technological tools, which can be like starting from your simple X-ray to advanced imaging technologies nowadays available. Why does the diagnostic matter? Because it enables early detection of the diseases. It could be like infectious diseases, or this could be also lifestyle diseases, all right, or genetic diseases, all this. So we can guide the treatment decision, and also we can explore the antimicrobial stewardship, and it also supports surveillance and outbreak response. We are all from Southeast Asia. You know that we always have so many outbreaks, right? We have SARS-1, then COVID, then we also have avian flu, bird flu, and on and off dengue is also very common in Singapore. So these are all some of the examples that I can give you. And also, if you are able to detect the disease early, then the healthcare cost may be lower and also the disease burden. Now, if you look at this picture, at any point of time, if you think I'm just going fast, or if you're not able to understand, you can just unmute the mic and talk to me. So, if you consider the diagnostic in 1980s or before 1980s, these are the gold standard method, culture, bacterial culture. You have a wound which is not healing, or you have a urinary tract infection which does not treatable by some antibiotic. Then what we do? We culture the specimen sample. Then we identify what kind of bacteria they are, what kind of organism they are. Then we try with the different antibiotics, culture techniques. It is very old, more than 56-year-old, but still we are using it. Then gram staining, we're still using the gram staining. In fact, in microbiology class, our student used this gram stain technology to classification of to understand the type of bacteria. Biochemical, even though they are all old, biochemical, serology, blood work, they are all old, but still we are using. So the point is, even though there is so much advancement in diagnosis like real-time PCR imaging or artificial intelligence or sequencing, so much variation of PCR, we are not doing away with the older technology, so-called older technology. But what this recent technology, they actually complement the technology that we used earlier. If you look at this picture, from culture, gram stain, biochemistry, serology, then we move to ELISA and DNA probes, then comes PCR. Does anyone know who invented PCR? Anyone? It is okay to give the wrong answer. I will not be able to see your chat messages. You can just unmute the mic and if you know the answer. Yeah, Kary Mullis invented the PCR, for which he got the Nobel Prize, right? Then comes Sanger sequencing. I think this question is very easy. Who invented the sequencing? Sanger, Frederick Sanger. Okay, he also invented the technique for protein sequencing. He got two Nobel Prize. Okay, one for protein sequencing and another one for the DNA sequencing. Then comes the next generation sequencing. 30 years ago, if you want to sequence a human genome, it takes like $3 billion, human genome sequencing project. The funding was $3 billion. It took nearly about 25, 30 years. Now, we can sequence the whole genome of any individual within a day or two. And the cost is only $300 to $400. We can specifically find out the genetic diseases, DNA mutation. And simple example is whether you have a mutation in BRCA1 and 2, which may lead to breast cancer. So the technology has so much advanced. So what do you understand by molecular diagnostics? Okay, molecular diagnostic to define in a very broader term is a diagnostic test that assess the individual's health literally at molecular level. That means we are using certain molecule to assess the health status of the individual. I don't know why nobody wants to talk. Tell me, what molecule will you test to see whether we have diabetes? Anyone? What is the molecule we test? Anyone? Are you all with me, guys? We all have glucometer, right?
Yes.
Yes, glucose. We test glucose to see whether some individual has a diabetes or not, right? That is, it is. That is what molecular diagnosis is. So we check glucose or pregnancy test kit. Okay, you can test in the urine or blood. Okay, blood is very early and very sensitive, but you can also test in the urine. We are actually testing a hormone called chorionic gonadotropin. This hormone is secreted by only after fertilization. Then what about the COVID-19 test? Of course, the gold standard technology is PCR, but we also used ART kit, right? Antigen rapid test kit, which you can do at any place. It's a point of care test kit. What do we test there? Actually, we take the sample from nasal sample. Where if they have the nasal sample has a COVID-19 antigen, which is a protein, spike protein, then it react with the antibody and the membrane, LFA membrane device. So we are testing the molecule. We are looking for a molecule called spike protein. So molecular diagnosis, we are actually assessing the situation, detecting the disease, looking at a specific molecule, right? This could be a DNA, this could be RNA, this could be a protein or any other metabolite. Okay, that is what molecular diagnostic is. So why I mentioned about it, we can test DNA or RNA or proteins. Protein in cells, this could be a enzyme, or it could be a hormone, and so on. This field is very dynamic and transformative. That means we see a lot of changes very, very frequently. Lot of changes coming in every day. Okay. Why molecular diagnostic? What are the advantages? High sensitivity. One example, classical example I can give is during a pandemic, when we were required to travel, the most of the airlines and immigrations in various countries, they need the COVID-19 RT-PCR test result. Why they need RT-PCR? Why don't they accept the ART test kit, where you can buy from 7-Eleven or any supermarket and you can test at your home? Why don't they accept it? Because this is not sensitive enough. Say, for example, you have only like a 20 parasite, 20 pathogen in your sample. If the antibody in the device may not be able to pick it up, too low. The sensitivity is too low. It may be also not sensitive, also not specific. In sense, it can also pick up proteins which are closely related to COVID-19 virus. Say, for example, SARS or influenza, Ebola. These are all the viruses that all belong to the coronavirus. So they can also cross-react, right? I will explain this, what is a false positive and false negative, in subsequent slides. So high sensitivity, high specificity. So when you test for COVID-19, the test you are doing should test only COVID-19, pick up only COVID-19 virus, not SARS virus, not dengue virus. That is what called specificity. Instead of picking up COVID-19 virus, if the test you are doing pick up a dengue, and it is called a false positive. If it is called false positive. Or you have a very low number of pathogen, bacterial load or viral load, but your assay cannot detect it. Sometimes it gives false negative. You have a disease, but the assay is not able to pick it up. Another advantage of molecular diagnostic is a speed, right? We just give the sample in the morning, we get the result in the evening. Actually, technically, you need only 1 hour to do the PCR, but we cannot just run one sample. So usually the research diagnostic laboratory wait for like samples like at least 48 sample. Normally we do in the 96 well plate, 96 sample at a time. 94 sample and a positive and negative control, 96 well plate, at least 48, so that you can maximize the use of the facilities as well as the reagents. So it is speed. Unlike the culture, you need to wait for the 2-3 days. You do the blood test, again you need to wait for 2 days, right? I don't know about other countries. In Singapore, it takes about 2-3 days. And you go for imaging, again it goes to imaging is done by some like X-ray technician, or like a MRI technician or CT technician, but it goes to the radiologist. They need to read, they have to interpret the results, and it takes time to receive it. So the molecular diagnostic is quicker, right? In 2-3 hours, you can have the result, and as, of course, the physician can start the treatment based on the informed decision. So the infection, this could be molecular diagnostic, as I said earlier, can be used for both infectious diseases as well as non-infectious diseases, genetic diseases. If you have a mutation BRCA, which can lead to breast cancer, or if you have any genetically inherited diseases, your gene carries some diseases. All these, like say for example, like thalassemia or sickle cell anemia, all this can be detected. Okay, so the diseases that we can commonly use to molecular diagnostic diagnosis, like it could be like viral diseases, say for example, COVID-19 or dengue, influenza, hepatitis, chickenpox, right, varicella, right, many. It could be bacteria, tuberculosis, or UTI, urinary tract infection, which is very common infection. If you go to any clinic, any hospital emergency department, you will see a lot of kids having like a UTI, I mean like high fever. And the first thing they do is they take the urine sample and they do put the paper strip, and that will tell you whether you have a UTI infection. Okay, sometimes they do also culture test. Yes, STD, sexually transmitted diseases. These are all caused by bacteria. Fungi can also cause disease. Candidiasis, the skin, skin diseases. Protozoan disease, malaria, amoebiasis. If you eat uncooked food, it may also cause amoebiasis, malaria. The salmonellosis. Salmonella is, like if you eat salmonella infected chicken, the Salmonella, Campylobacter, these are all like bacterial disease. Salmonella, Campylobacter, Shigella, all comes from chicken and egg, uncooked meat. Okay, when you do the barbecue, if you are in a hurry, if you eat half-boiled meat, this can cause. Only if the chicken meat is infected with this bacteria. So now, whether you are dealing with the virus, or whether you are dealing with the bacteria, or whether you are dealing with the fungi, whatever the type of organism, including higher animals like a human, all of this biological organism, they carry genetic material. We call it DNA. We have DNA, but some organisms, some viruses, they also have RNA as their genome. I hope DNA is double standard. We all have, how many chromosomes we have in our cells? Anyone? I see one Samuel in my name here in my screen, Sam. I see only a few people. Yeah. Anybody? Nika? Anyone can give the answer. Charlene, Ansela, Risa, anyone? Okay. Yeah, feel free to unmute the mic and talk. So we have 46 chromosomes, right? 23 comes from our father and another 23 comes from our mother. Some organism, they also have virus as a genetic element, genetic material. Say for example, COVID-19 virus. So why we are talking about whether DNA or RNA? Because when you are diagnosing, DNA viruses are much easier to diagnose. For RNA viruses like COVID-19 virus, they need an additional step that I'm going to discuss. Does anyone can guess what kind of cells they are? Do you see a cell here, right? You see a cell here. What kind of cells? Anyone? You see a cell here, right? One cell. Here is the nucleus. There is another cell here. What kind of cells there? Anyone? There is also another one more cell actually, which is actually squeezed here. There are 3 cells. Okay. Okay, guys. So these are actually cheeks. You can take the toothpick or cytological brush. You can extract. Take a swab from your cheek inside. Okay. Then you can just make a smear, and then you put a toluidine blue. Then you can observe in the microscope. You can see yourself. Of course, this is my own cheek cell. During the cell biology lab, I just took this picture. So you can see here, this cell, the cheek cell, is around like 60 to 80 micron. Because when you make a smear, it can also elongate. Okay, so do you also see some tiny, tiny spots like this here? Tiny, tiny spots. What are they? They are bacteria. Because when you eat, you have a lot of food for the bacteria. The bacteria grows. Okay. So the intention, the point for showing this picture is that you can compare the size of the bacteria, human cells around the 80 micrometer, and the bacteria is about 1 to 3 micrometer, and the viruses are only 0.32, 0.032, 0.3 micrometer or nanometer, 30 to 300 nanometer. They are very, very small. You cannot see by microscope. That is why I use the term invisible. So how do we diagnose? We do PCR or culture, or look at the antibody, immune response, and so on. Moving on. So I would like to mention about this. When SARS, that the SARS actually originated from Wuhan in China. It quickly spread all over. Okay. And Chinese scientists sequenced the genome of this coronavirus 2. The SARS stands for Severe Acute Respiratory Syndrome Coronavirus 2, SARS-CoV-2. Okay. So the Chinese scientists very quickly sequenced the genome and shared with the Singapore scientists. Singapore has a bioinformatics institute. They shared the genome data, the genomic sequences, and they actually helped to design the primers for the PCR diagnosis. So I'm not going to discuss on the classification of viruses, but this virus that we are going to talk today is a single-stranded RNA positive virus. Later, if you have queries, we can discuss on that. Now, since we are focusing on the pandemic, so this virus is RNA virus. So you collect the sample, right? You collect the sample. I mean, you go to the clinic, the nurse or physician collect the sample and they send it to the diagnostic laboratory. They don't do that there. Of course, if you go to big hospital, they have a diagnostic laboratory in the close by. Most of the cases, if you are going to a clinic, a small clinic, they collect the sample and the delivery guy, they pick up and they deliver to the diagnostic lab. They do batch by batch. So during this time, your sample need to be protected. Okay, number one, it has to be properly stored. It has to be quickly transported, right? Then they isolate the RNA, because your RNA is inside the cell. You have the cell membrane. The virus is already inside the cell. In order to diagnose, in order to do the PCR amplification, you need to break open the membrane, release the genetic material so that we can make copy of it. So basically in PCR, we are going to make copy. That is what we are going to do. I will come to that later very quickly. So we need to extract the RNA, purify the RNA. What do you mean by extraction and purification? Break open the membrane. There are plenty of stuff. You have proteins, you have carbohydrate, you have lipids, you have minerals, vitamins. Then the nasopharyngeal sample has saliva, right? Saliva that is very viscous. Okay, they have mucus. All this will interfere with the polymerase chain reaction, DNA amplification process, RNA amplification process. You need the clean DNA or RNA. That is what we do. But there is a problem with RNA. We all know that in the cell, in the nucleus, we have chromosome, which are actually DNA. DNA store the genetic material. Say, for example, melanin, the skin tone you see, it is actually by this. Why some people are like dark skin, some are like white and so on. It's all how much melanin is expressed in your skin. So we all have the same DNA, but how much is expressed? Say, for example, the DNA is transcribed, rewritten in the form of RNA, which then transported to the cytoplasm. In the cytosol or cytoplasm, RNA is translated to a protein, a pigment called melanin. So, how much protein are synthesized is very important. How much protein are? So, how many copies are made? You have only one copy of the DNA. How many mRNA copies are made? It depends on so many factors. Then how long the mRNA stay inside the cell? Because this can keep on translating multiple times, again and again and again. You will have like millions and million copies of melanin or amylase. You take rice, then you need amylase to digest. So this is what happened in DNA, DNA to RNA, RNA to protein, like transcription and translation. So, but unfortunately, the RNA are very small. DNA is very long, double-stranded. It is protected with so many proteins, like transcription factors, DNA polymerases, single-stranded DNA binding proteins, hundreds of proteins. So DNA is very highly protected. Even otherwise, DNA, even from like a Stone Age, you have the DNA from some fossil sample, we can still do PCR. But unfortunately, RNA can be easily degraded. That is a problem with diagnosing RNA viruses like COVID-19 virus. Why RNA can be easily degraded? Because RNA is short. The chromosome number 1 itself is 250 million bases, ATGC, nearly 250 million, 248 million bases. But your RNA, COVID-19 major spike protein, 300 amino acid, that means only 900 bases. That means not only they are shorter, they are also single-stranded. DNA is a double-stranded. It is protected with so many proteins, but your RNA is single-stranded. Okay? Now what will happen? The enzymes, RNases, can rapidly degrade from both sides, exonucleases. They can degrade your RNA. So if the sample is not protected, your sample is not properly prepared in the lab, your RNA will be degraded, then you get a false negative result. That means you have the disease, but unfortunately, during the sample preparation or storage, the viral RNA is degraded by enzymes. The enzyme could come from our own cell, the nasopharyngeal cells, right? It could be also from the environment, because the environment is very rich in E. coli, common E. coli. This can come from the equipment, glasswares. All this will degrade. That is why we need to keep certain practice, like wear glove all the time, use RNA-free tubes and pipette tapes, use dedicated space and chemicals, okay? Supplement the reaction with RNase inhibitor. All these are some of the precautions that we take for the RNA work. So I just want to give you some idea. I think the COVID-19 was reported by Chinese government in October 2019, here and there few cases. In Singapore, the first case of COVID-19 was reported, if I'm not mistaken, January 21st, 2020. Okay, January 21st, 2020. That was the first case. Okay, then that during February, first week of February, I was doing a course in National University of Singapore. We were like wearing masks, but like those days, like the number of cases in Singapore is like 7, 9, 10, like that. We never thought we would face such a like lockdown and serious consequences, all this. Nobody would have thought. But at those days, we were actually, Singapore scientists were doing the COVID-19 test manually. They extract the DNA, RNA viruses manually, manual method. You like grind the tissue, macerate the tissue, homogenize the tissue, then add the binding buffer, wash it, then wash, elute manually. Because you have only few samples, 100 sample a day or 200 sample a day. We were able to do only 300 samples those days. But suddenly, because of the rapid spread of the disease, then we were forced to do like hundreds and thousands of tests. Then comes the automated machines. These automated machines that I'm going to show you very quickly, quickly, these are the machines which can actually prepare the RNA from the sample, 96 sample in 2 hours. It not only prepare the RNA, but also set up the polymerase chain reaction. Okay, all these everything is automated. Why automation is important? Because you can handle large number of sample. From 300 sample a day, Singapore was able to do 100,000 tests a day. Even Singapore had a laboratory in the Changi airport. Okay, all parts of Singapore, we had capability to diagnose 100,000 sample a day. Don't compare with China, Philippines, or India, which handle like 10 million sample every day. But Singapore is a very small country with only around 5.67 million population. Okay, so this method, the automatic method, uses the magnetic beads. I don't know how much time I have. I think I have another 30 minutes. So I will skip this part. Later, if you have any questions, we can discuss more on that. So you would have also heard from the hospital, the nurses and doctor getting infected. The technician working behind the scene. They are the warriors. They actually work 8 hours, 10 hours, 12 hours, even 16 hours without sleep. Some doctors 24 hours, right? During the pandemic. So they were some heroes. They were working behind the scene. They are all like the medical technologists, medical laboratory technologies who continuously test our sample. They may also get infected. Of course, many were get infected. So, and also when you handle like 100 samples simultaneously on the desk manually, there is a possibility for contamination, cross-contamination. Okay, so you can also handle large number of samples. These are advantages of automated workstation. So the FDA actually authorized some of the machine, and this machine is called COBARS. It is a French abbreviation. This machine, the beauty of this machine, it costs several $1 million. Of course, in some of the laboratory here in Parkway Laboratory, our DSO, Defense Science Organization, all these laboratories had this machine. This one, you sample in, result out. Okay, one machine can handle about 2,000 sample a day. So you put one batch of sample, 96 sample, RNA is prepared, then it goes to the PCR stage. When it goes to the PCR stage, you can put another tray with 96 sample to do the RNA extraction. Like that, you can do 8 or 9 cycle. So you will be able to diagnose, test around 2,000 samples, sample in, sample out. But this machine is expensive, probably like a few $1 million. Then these two, this is one of the machine we used in our Temasek Polytechnic. In Temasek Polytechnic, we are not the testers, but we train the COVID-19 testers. I was the lead trainer for this program. We were approached by Ministry of Health and Ministry of Manpower to come up quickly with a course, short course, to develop the capability to train the COVID-19 testers, because there was a huge demand. You know that Singapore is a small country and we have a lot of people traveling from Malaysia every day for work. You just need to cross the bridge. Every day, approximately 200 people cross the border to come to Singapore to work. Okay, so when the border is closed, we have also neighboring country Indonesia, just on like 30 minutes, 45 minutes, we can reach Indonesia by boat. But unfortunately, when the border is closed, we don't have that much workforce. So we have to train the local talent, right? So we trained about 250 people. Then they were actually involved in COVID-19 testing. So this is actually, these are the machine provided by the Singapore government to train the COVID-19 testers. This machine is called PreNAT. Pre, you know the meaning. NAT stands for nucleic acid test. Nucleic acid test. Okay, nucleic acid, NAT stands for. We still have this machine. Another machine is from, I am not going to tell the name. I'm sorry. Do you guys hear the volume, sound, audio? Luna?
Please play it. Yeah, please play it. Oh, no, here's no voice.
Okay. In that case, I'm sorry, I have to stop and my apologies. I have to stop sharing. I have to reshare the video again. Slide again. Sorry about it. Okay. I need to click this share sound. Then go here. I'm sorry. We don't use Zoom. I have not used Zoom for 4 or 5 years. We use Microsoft Teams, so I'm not very familiar. So let me go to the video.
Yeah, no worry.
This machine is from Wuhan, where actually probably the first infection was reported. Okay, this machine we bought for about $200,000. Okay, you can convert to your own currency. That is a lot of money. So it is a beautiful machine. You can see here. Yeah. So this machine actually prepare the DNA. It actually can do 2 plates simultaneously, 96 plus 96, 184 sample at a time, including the positive and negative control. Why do we, as I think I mentioned about this, why do we need this nucleic acid test? Because your antigen rapid test kit may be not sensitive enough, also not sensitive enough in sense, if you have too low, antigen is too low virus, number of virus is too low, it may not be able to pick up. It gives you false negative. False negative. That means you have the disease, but the test is unable to pick it up. Sometimes the antigen that you take from your nose, it can also react with other antibodies in our system. Say, for example, you were previously infected with other similar closely related pathogen, it can cross-react. In that case, it will give you false positive. Both are dangerous. When you say false negative, you may have the disease, but it says no disease. you are not going to take any treatment and you are going to spread the disease to everyone. In the case of false positive, okay, you unnecessarily traumatize a person, right? He doesn't have a disease, but he is mentally disturbed, stressed, that feeling that he has a disease, right? So these both are dangerous. Just to give you some example, Government of India purchased nearly 500,000 COVID-19 test kit, ART test kit from China. But unfortunately, because of the false negative, false positive problem, they actually canceled the order. And actually, yeah, so this is all happened like during the COVID-19, but this is it is not anybody's fault because this assay has a limitation. All right, that is why. So here is a simple picture that tells you what is a false positive and what is false negative. Here you don't have a disease, but this picks up, yeah, you have a disease because, say, for example, it pick up a wrong pathogen. So it is false positive. Similarly, the false negative. And that is why the WHO suggested to do the nucleic acid test for the COVID-19 diagnosis. And what is nucleic acid amplification is, we are going to, I mentioned about the DNA and the RNA, and this COVID-19 virus is RNA virus. It has 30,000 bases, sorry, not base pair, only bases, because it is single stranded, right? 30K bases. So we are going to pick up a sequence around 100 bases, which are very unique, present only in COVID-19 virus, not present in any closely related. So I think I have deleted the picture or something. Maybe it is coming later. So these are the target: N gene, envelope protein, and spike protein, and RNA-dependent RNA polymerase gene coding for this. These are the 4 targets suggested by the WHO. So normally, if you have the DNA virus, you extract the DNA and amplify the DNA copies. Because in the ART kit, you have only 5 copies of the spike protein. It may or may not be able to detect. But in here, even if you have 1 copy, you are going to make copy of it. Polymerase chain reaction. That means it is not like making a photocopy, but here, how does it work? 1 become 2. Then the 2 become a master copy. So 2 become 4, 4 become 8, 8 become 16. And this happens, this happens for about exponential amplification take place about 40 to 45 cycle. But in the case of RNA virus, unfortunately, you have a single stranded RNA, which cannot be directly amplified. Of course, there are technology to amplify the RNA to RNA, but here, remember, we are going to use a fluorescent dye to indicate the amplification in real time. That means in real time, we can see whether there is any amplification. So there is an additional test involved, additional step, which is called RT, reverse transcription. We convert this RNA to DNA, double-stranded DNA first, then we amplify them in the presence of a probe, a color probe. Okay, and this is the 30,000 bases where we are going to use the spike protein mainly, and these are some of the targets suggested by the WHO. I'm going to skip this slide. And you also know that, unfortunately, like COVID-19, any organism, our genome constantly undergo mutation, like a physical phenomena, like even if you're exposed to sunlight, especially UV light, our DNA, some chemicals, food habit, many things alter our DNA, mutation. So older the individual, more mutation we have. But they are not heritable unless this mutation happens in the sperm or egg cell. So this virus also underwent like a lot of mutation. The first mutation was reported in December 2019, sometime, here. December 18, 2020. In this, so what I had some students, some are doing summer project. We come up with a simple technology, because if you want to detect the mutation, you have to sequence the whole genome, right? But we come up with the technology using high resolution melting, where we amplify the gene, then melt at high resolution, melt slowly. Because of the mutation, because of the G changed to A or T, G and C, and A and T. G and C in DNA has 3 hydrogen bonds. They melt at high temperature. A and T, only 2 hydrogen bonds. They may melt a bit early. So that is what we are detecting here. The wild type melt at 70, 5.5, and the mutated one, alpha strain mutant, they melt 1 degree early. So this is the technology we developed, but unfortunately, by the time my student Alexander, Daniela, and Clara, they developed the technique, we had a few other mutations. So the mutation was faster than the technology we developed. So then actually it is a good learning, but this technology is useful for diagnosing, detecting the mutation. Now, we talked about the PCR and PCR. I'm going to show a short video to explain what it is.
There was a time when the amplified DNA,
you had to grow tons and tons of tiny cells.
Ooh, and along came a guy named Dr. Kerry Mullins.
Said you can amplify in vitro just as well.
Just mix your tablet with a buffer and some primer. Nucleotides and polymerases too.
denaturing, annealing, and extending.
Well, it's amazing what heating and cooling and heating will do.
PCR, when you need to detect mutation. Detect mutation. PCR, when you need to recombine. Recombine. PCR, when you need to find out who the candy is. PCR, when you need to solve a crime. Solve a crime. PCR, when you need to detect mutation. Detect mutation. PCR, when you need to recombine. Recombine. PCR, when you need to find out who the candy is. PCR, when you need to solve a crime. Solve a crime. PCR.
So this is actually a commercial ad, 25 years old ad for the PCR machine. But this song, PCR song, I call it, as well as the music actually teaches the basic concept as well as the applications of PCR very clearly. To summarize it, what PCR is, I mentioned imagine you have only one copy of the DNA, only one copy of the DNA, which may not be, which we may not be able to detect, right, from the sample. In the PCR, we are going to make a copy of it. It is actually what happens in our body. Say, for example, you have a small wound, you have a small cut. When you are doing helping in the kitchen or doing some gardening, imagine you have a small cut. What happens? The wound heals in few days. There is a generation of new cell. Before the cell divide, the new cell and the older cell all have 46 chromosomes. That means it divides. The DNA replicate. DNA division, that is called replication. Okay? That happens naturally in our body. Every organism have this property. The same thing we are doing in the test tube. It is actually in vitro replication, DNA replication. In body, there are a lot of enzymes, proteins to separate the DNA. to synthesize the primer, then extend it. So a lot of process. But in a PCR tube, we minimize everything. Instead of using the enzyme, we raise the temperature to 95 degree. At 95 degree, what will happen is the two strands of the DNA will separate. Now, the short stretch of nucleotide, short stretch of nucleic acid called oligo or primer, they are complementary to the strand. You have a, the primer will have T. You have G, you have a C. So we design it. How do we design? Remember, I told you when the Chinese scientists sequenced the COVID-19 virus, they shared the data with the Bioinformatics Institute Singapore scientists. They together designed the diagnostic tools like primers and so on. So this data is available in the public domain. You can just go to the Google and type GenBank, okay, GenBank, okay, or European Bioinformatics Institute. All this data from human, plants, animal, microorganism, all this you can get. So based on that, you design this primer, which are complementary. So at this temperature, you lower the temperature to 45 to 50, which depends on the sequence of the primer. Not all PCR are same. Every PCR is different. You need to design, program it accordingly. Then what you do, you raise the temperature to 72. Now, there is a special enzyme called Taq polymerase. We cannot use human polymerase or E. coli polymerase because remember, we raised the temperature here to 95. If we use a E. coli polymerase or a human polymerase, the enzyme will be denatured because it's protein. What happens when you boil the protein? The egg white is denatured, right? It actually use the egg white, you can see what happens, right? So similar thing will happen here. So we have a special polymerase called a polymerase, DNA polymerase obtained from Thermophilus aquaticus, a bacteria that survives in hot spring. Okay, it can survive at extremophile, thermophile. It can survive at very high temperature. So this enzyme is from there. So what this enzyme will do is it will extend the primer. How does it do? The same DNA replication principle that happens in our body. You have A, the Taq polymerase will pick up the T from that we provide and add it to the primer. You have G, it pick up C. You have G, you pick up C and add it because G and C and A and T make pair in the DNA. So that is how the extension take place. Now, one copy become two copy. What do we do now? Then we start again. So 2 become 4, 4 become 8. You do this for 40 times. If you are good in mathematics, you can calculate with one copy how many copies you will get after 40 cycles. That is what happens in the PCR. But how do we know whether there is an amplification? Normally, when we do in the PCR, I know some of you may be taking biology. You do electrophoresis, right? You do electrophoresis for 10 sample, okay. 20 sample, okay. But we have like India on average, it's nearly screened about 1 million sample a day, right? 1 million sample, 2 million sample every day. Probably China as well, big countries, right? You cannot do electrophoresis. They need some advanced technology without involving so much labor and manual work. So that is why we use a real-time PCR where we are going to use a dye. So when there is an amplification, which can be captured by the camera in the PCR machine, which we can visually see without any further experiment. So I mentioned about why RT-PCR. Even though it is called a PCR, it is not a cyclic reaction. Imagine your sample has 100 viral RNA. It is just one time, okay, one time. At the end of the reaction, 20 minutes, you will have probably 70 to 80 cDNA. Considering your assay is 70 to 80% efficient. Okay, it is not going to make 100 RNA to 5,000 or 10,000 RNA. Okay, it is the first step, RT, reverse transcription. Then after reverse transcription, I'm going to skip this slide. After reverse transcription, Now you have the double stranded DNA. We are going to do the amplification now. In the amplification, the PCR, we are going to use a probe. A probe, later I'm going to see the picture, a probe, which is actually, I can draw here, a short stretch of nucleotide like DNA. On 5 prime, you have a reporter, a fluorescent dye. On 3 prime, you have a quencher. You have a quencher like Iowa black. Here you have a Sci-Fi or VAC or Hex and so on. What will happen? This is only like 10 to 15 bases nucleotides, very short. They are in the very close proximity. So what will happen? This fluorescent reporter will continuously emit fluorescence because it's fluorescent, right? But what will happen? Because the quencher is in the close proximity, it will quench the fluorescence. It will absorb the energy. This process is called fluorescence resonance energy transfer, FRET, physical phenomena. So what will happen when the two molecules are in the close proximity? When a molecule excites the energy, the other molecule absorbs it. So here the fluorescent energy, light energy is absorbed by the quencher. So you won't see the fluorescence. You won't see the fluorescence. But how do we use this in our PCR? Remember, guys, I mentioned about an enzyme called Taq polymerase. This enzyme, Taq DNA polymerase, not only extend the primer, not only extend the DNA, it also has additional activity, which is called 5' nuclease activity, which means the DNA is 5 to 3. Whenever it finds a DNA, it starts from chopping off the nucleotide one after another. A nucleotide, a DNA, ATGC or deoxyribonucleotide, they have a sugar, they have a nitrogen base, and they are connected to each other by the phosphodiester bond at the 3rd carbon and the 5th carbon. The 5 says 5th carbon, okay? The carbon 1, 2, 3, 4, and 5, CH2OH, sorry, CH2OH. So here is a 3rd carbon a bond and the 5th carbon a bond. This is how the DNA chain looks. So this enzyme activity, so the Taq polymerase has inherent property of this nuclease activity, which will chop off any DNA, single-stranded DNA, one after another. So now you know what is a probe. Now you know the activity of this. Now you will easily understand how does this real-time PCR work, unlike the regular PCR. Now, imagine the sample you have is positive. You have the DNA here. Okay? Your primer, forward primer binds. Your reverse primer binds. Your forward primer, you just imagine 1 to 20 bases. Your reverse primer, 100 to 80 bases. Sorry. So you can also, you have to design your probe between 21 to 79 bases. You can design anywhere, which are also complementary to the target you want to amplify. Okay, now when you start the reaction, what will happen? The polymerase, DNA polymerase, will keep on extending it, adding the new bases. But when it reaches this, remember it has a 5' exonuclease activity. It will start chopping off the DNA, the probe. Remember, earlier, this is actually the fluorescent reporter and the quencher are tied up together. So all the fluorescence were absorbed by the quencher. Now, they are no longer tied up. They are no longer in the close proximity because the nuclease actually broke it so that it can extend the PCR. Since they are no longer in the proximity, now we can see the fluorescence. Now, the fluorescence can be detected. So there is a camera in the PCR machine, optical detector, that will detect the fluorescence and transfer to the computer or inbuilt device, what you call a visual display where we can see. That means you will see the fluorescence only if there is a positive sample PCR amplification is taking place. If there is no DNA, viral DNA, no PCR, you will not see any fluorescence. Let me show this short picture, short video. Yeah, probably I
This
will
program
need, probably I
shows
will wrap
how
it up in 5 to 10 minutes maximum.
a specific nucleic acid in a clinical sample ingredient labeled covalently with a fluorescent molecule, and another nucleotide is labeled with a fluorescence quenching molecule. The quencher rapidly absorbs any light energy emitted by the fluorescence molecule as long as it remains in close proximity. Now, let's look at what happens when this additional ingredient is present during a single cycle of PCR. As the primers bind to the separated strands of DNA, the probe also finds its complementary sites between them. The enzyme that synthesizes new DNA from the ends of the primers also has a second activity, an exonuclease activity. So when it encounters double-stranded DNA in its path, it will disassemble the strand that is in its way and replace all of the nucleotides. As the polymerase cuts through the probe, note that the nucleotide bearing the fluorescent marker and the one bearing the quencher are separated from one another. In the absence of a nearby quencher, the fluorescent molecule can now emit detectable light when stimulated. Each time another amplicon is produced, another fluorescent marker is released from its neighboring quencher. Therefore, just as the number of amplicons doubles in each PCR cycle, the amount of emitted fluorescent energy also doubles. This light generation can be monitored during the PCR reaction. The probe also finds its complementary sites between them. The enzyme that synthesizes new DNA from the ends of the primers also has a second activity, an exonuclease activity. So when it encounters double-stranded DNA in its path, it will disassemble the strand that is in its way and replace all of the
Okay, guys. So as I mentioned, so there is a camera fixed in the PCR machine, which will actually, you can see here from 1 cycle to 35 cycle. So it can actually pick up the fluorescence. We can see this fluorescent peak. So if you have a very low sample, very low viral DNA, say for example, 1 copy, you will see this here after 30-35 cycle. Your sample has a lot of viral load, like 100,000 or 1 million. So we see it very quickly. So now the machine will draw a line, and wherever this the fluorescent peak crosses the fixed threshold, we call this cycle, cycle number as CT value. If you have a question, I will explain later on the CT value. Normally the machine build it, and based on that, we can actually use a Excel file to calculate, or you can also manually calculate. But sometime, remember, the CT values between 18 to 35, you can calculate the copy number from the graph, which is called interpolation. Imagine if the CT value is 7 or 8 or 9 or 10 something, that is called extrapolation, right? Use the formula, which is actually y is equal to x minus b. That is what I have shown in this graph, okay? But in our case, we know the y, but we need to calculate the x, okay? This is a very simple calculation. You don't need to do manually because the machine will do everything, but for you to understand. So the point here is, lower the CT value. If your CT value is like 5, 6, that means your viral load is very high, very high. And if you have like your CT value is like 28 or 30, your viral load is low. So why the CT value is very important? To see the viral load, whether a person is improving, COVID-19 person is improving, not only applicable to COVID-19, You discover a new drug, you treat the patient with a new drug, you can see whether the patient is improving, your drug is working. So it has a lot of applications. So lower the copy number means higher the severity of the disease. This is applicable to any kind of diseases or non-infectious or not infectious, many kind of biological studies, not only for COVID-19. So with this graph, you see it is called a semi-log paper, semi-log graph. You have the log scale on the x-axis and linear scale on the y-axis, semi-log, because your increase, the amplification is, every cycle is exponential. So that is why it is. So we can use the formula to calculate. Here is the video. Probably I will send this video how to operate the machine. This is the machine, PCR machine. Yeah. I think I will quit this. I think
And they are called FAM.
I think this is how to program the machine. I will skip it. If you guys need, I can send the video to Miss Luna or Ifan, and they are free to share with them. My last part is the actual work that I'm currently doing. But unfortunately, I think you are all muted. Probably some of you are messaging me, but I'm not able to see the message at this point of time. So I don't know. I think probably many of you know who they are. They are Nobel winners. Jennifer Doudna and Emmanuel Charpentier. Jennifer Doudna is from University of Berkeley, University of California at Berkeley, and Emmanuel Charpentier is from Europe. They actually made a discovery that the CRISPR enzymes can be used to precisely edit the gene. All right. Of course, CRISPR was discovered a long time ago by Japanese scientists in the 1980s. But these scientists, Professor Jennifer and Professor Charpentier, they showed that these enzymes, Cas9, can be used to edit the genome precisely, which are already in the therapeutic area where you can cure thalassemia, sickle cell anemia, night blindness, and so on and so on. Several genetic diseases can be cured. In fact, probably those who are from China probably know that 3 or 4 years ago, one Professor He Jiankui, he actually produced 2 babies, Lulu and Nana, who actually modified the genome. He edited the embryo because the father had some genetic diseases. He doesn't want, the family doesn't want the chip to inherit. So they approached the professor and he actually edited the embryo genome. And they have 2 CRISPR babies. We don't know the name. We just only know that they are doing fine. So for this work, we have the technology. I think, yeah, he was arrested and he was in jail. And more recently, we know that he is also released because it is not ethical to edit the human or clone the human babies, genetically modify the whole babies or embryo. So the CRISPR also has additional property which we can use for CRISPR-based diagnostic, but for want of time, and I will not elaborate. Later, if you have some question, I can do that. So we use CRISPR technology to diagnose the diseases, which are not our invention, but they are all invention from 2 laboratories from MIT and Professor Jennifer Doudna's lab. But we have made a 3-in-1 multiple recombinase polymerase amplification. You can actually detect this without the PCR machine because PCR machines are expensive. You need technically skilled people. And we have also used this technology to develop, to identify antimicrobial resistance. But unfortunately, these are all involved like handling small volume of the reagent. So we have also developed a microfluidic device, which we can actually close the system where you can just put the drop of the sample and identify. These are all still in the prototype level. We will be able to commercialize it probably in around 6 months time. So I just also want to mention that, you know, our approach is One Health because human, environment, animal, we are all interconnected. Many of the diseases that we have today, 60% of the disease actually come from animals, like COVID-19 came from bat, like we had like zoonotic, many zoonotic diseases, like mad cow or avian flu or swine flu, all comes from animals, right? Because we share the common environment. So the approach is at One Health, where we have to continuously monitor the environment, human, and not only the pet animals, but also the farming animals like cattle, aquaculture, and so on and so on. So our work currently focus on developing a CRISPR-based technology which can be used for the One Health diagnostic. So what is the future of diagnosis? So we actually started with the cell like culture and serology or chemistry, but now lot of advancement like CRISPR-based technology, lot of advancement in imaging, and no need to mention that also the AI, artificial intelligence, play a major role in diagnostic, especially in data analysis from like imaging system, comparing like thousands and millions of images, and give you some informed decision to make. And also many of the workflow are automated and it generates a decision report. So some of the suggestions for physician to do, and also integrate the technology to like wearable devices, even access your smartphone. All this data are another area of interest. And so the ultimate take home message is when you have a disease, most of the time we treat the symptoms. Say, for example, you have a headache, and normally we just take a Panadol. Panadol actually does not treat the disease. It actually treats the symptom. You have a chest pain. Most of the time, it is going to be like a indigestion or a musculoskeletal, but it could be also serious problem like heart attack. So it is very important to diagnose the disease, understand the root cause of the disease. This one has been said by one of the Tamil poet. I originally from India, from Tamil Nadu, but I moved to Singapore a long time ago. So this poet says, "Noi nadi, noi mudal nadi, adu thanikum vai nadi vaipache." That means disease, understand it, the cause, whatever it is, then examine, understand the root cause of the disease, then you start treating it. That is what the basic of diagnosis. And this is the picture from COVID-19 4-5 years ago, some of my students. And I will stop here. And here is my business card. You can just scan it. And here is my LinkedIn. And here is my email. And feel free to contact if you need any help. Yeah, I will stop here and leave the floor to Ms. Luna and Ifan and her colleagues.
Thank you so much for engaging in Riching's presentation. So next we can move to the next section. Okay, and yes, and now we're in the Q&A section. If you have any question, you can just type in the chat box or open your microphone to ask your question. And as for now, we already see some question in the chat box. So Dr. Rage.
Now, give me some time. I'm still struggling to go back to my Zoom. I don't know where I am. Zoom. Okay.
Okay, they have several questions. Please answer it one by one. So the first question is from the Vincent.
Okay, give me 1 second, Luna. I'm still struggling to.
Okay.
Yeah. Okay. I don't know how to maximize the window. Okay, it disappeared again. I'm sorry, I have not used Zoom for a long time. Yes, I can see here. Yeah, chat session. I cannot see. Okay, good morning. Okay. When webinars will be end? You are free to move, man. Why this question? Or this presentation slide shared in the WhatsApp group. I will give you the edited slides. Some of them are like we may not have the copyright. I will send you a set of slides for your use.
You can answer the question about the presentation first.
I'm sorry?
Yeah, you can answer the question about your presentation first. And here
I don't know where they are. Yeah, okay. Can you help me to read this? Yeah.
Yeah. So for the first question is from the Vincent. Vincent Jace. Yeah, the question is, given the speed of the molecular diagnostic innovation during the COVID-19, how can we ensure the same possible faster emergence and accessibility for others engaging disease before they become a pandemic?
Thank you, Vincent, for the question. Yeah, I'm able to read now. So, how can we ensure the same or possibly faster urgency and assess the other emerging disease before they become pandemic? So that is the I had the last slide answer the question. We need the continued surveillance. It is not only we develop something when something comes up. We continuously, the government agencies, they continuously monitor the environment. Okay. Environment, human, animal, we need to continuously monitor. There should be also intergovernmental cooperation to do that. So we can predict early and we can be well prepared rather than something to break out. Then we look for the solution. So why are other people doubting the DNA results? Is there a possibility of altering the result? Why are other people? I don't know who doubts the result. I don't understand the question, Samuel. It depends on the place. I don't think anybody can, hospital, it depends. Some countries have a very strict guidelines. There is no need to doubt DNA results unless there is some sample mix-up in the laboratory during the procedure. As I mentioned, quality of the sample is very important for the accuracy of the result. If samples are not properly prepared, properly stored, the experiments are not properly conducted, the results may not be reliable, but we have the quality control for everything. Even for COVID-19, we use 3 different quality control, okay? So I don't know why you asked this question. Sometimes it happens the samples are wrongly labeled, or sometimes the samples may be denatured, spoiled because of the storage condition or extraction or contamination and so on. But DNA results are highly accurate, okay? Whether it is a forensic or genetic testing or it is not. DNA tests are not only for diagnostic. These are the common techniques the forensic scientists they use. Say for example, if you obtain a blood or urine or semen or even piece of tissue or hair obtained from a crime site, they do the DNA fingerprinting, which is similar to the PCR. Basically, it is a PCR technology. Okay. So with that, only like hundreds and thousands of crimes are resolved. In the DNA database, the government of US maintained the DNA database. Even for parking efforts, they take a sample. Within 1 hour, they can identify your history. I think the last time I checked it they had more than 3 million. 4% of the US population data is already there. Okay, but I don't know any other countries have this DNA database. So DNA data are highly reliable unless somebody tamper with your results. I don't think it happens. I have watched some movie where people actually tamper the DNA results. I don't know whether it happens in real. Okay. I don't know how much time we have, or maybe I can give some example, Luna.
Yes, please.
In one of the movies I watched, one of the movies, it's not a real. I can also tell you a real example, okay? Say, for example, in Germany, several years ago, they found a DNA sample from the gloves of the robbery, money heist, right? Bank robbery. They had the DNA sample. They know the person who it is, but unfortunately, he has identical twins. Identical twins come from the same embryo, right? They have the same DNA. So they could not convince clearly that who among the 2 twins was involved in the robbery or heist. So the judge has to release both of them. It happened in Malaysia also, 2009. The drug trafficking case, similar. Both of them are released because the philosophy is the innocent should not be put in jail, right? So, but now technology has advanced. We have the whole genome sequencing. even though they come from the same egg, right? During the growth, our DNA undergo a lot of changes, right? Mutation I mentioned. You can sequence both of their genome, compare with the one from the crime site, we can compare. So genetic testing, number two is genetic testing. For want of time, I do not want to elaborate. You can just go to Google and type baby P, where somehow the sperm is mixed up. This is a very interesting case that happened in Singapore 10, 15 years ago. Okay. Okay, let me move to the next question. I hope, Vincent, I have answered your question. There is my project research on mutation spectrum, tubby-like protein patients in. So I'm trying to know how many genes are to be involved. Some of the diseases are caused by single gene, single mutation, one base, thalassemia, sickle cell anemia, one base. Some of them are complex, so it depends. You need to do your own research. Say, for example, autism, many genes are involved. What is the role of molecular biology? Molecular biology and molecular diagnosis are same. Molecular diagnostics is a set of molecular biology. What are the what do you mean by three molecular diagnostic technique? There are like several molecular diagnostic technique. PCR is one, Western blot, ELISA, even glucometer. These are all you are testing the molecule, right? Then you can also even with the imaging, you can look for certain molecule, labeled molecule we can check. There are many, it's not limited to three. Ansela, how do molecular diagnostic help in monitoring and controlling food safety? Very interesting question, Ansela. Actually, I work on the food testing as well. So, in Singapore, we import like 90% of the food we import from overseas, more than 20 or 30 different countries. Rice we import from China, Philippines, Thailand. Beef we import from Brazil, Australia, beef, pork, vegetables from Malaysia, India. Unfortunately, from Philippines, government wanted to import some chicken and pork. I think that during that time, there had some swine or some swine flu, so that did not materialize. So for Singapore, food safety is very important. Every day, Singapore laboratory, the Singapore Food Agency performed 1,000 tests. Technically, I'm telling, because last year they performed 340,000 tests. Okay, everything, food, even the milk powder we import, everything is being tested for chemical, like fertilizer, pesticide, and also biologicals, like we import shrimp from China, Indonesia, even from Thailand and many countries. We look many pathogens by PCR and many other methods, because even though these pathogens are not infectious to human, because when you cook, the DNA will denature. When you eat the DNA, your enzymes are going to degrade. But for biosecurity reason, we don't want these diseases to spread in the local farm. So the Singapore Food Agency, they do take care of this. Of course, the technology they use is real-time PCR. In fact, we had a project with the Singapore Food Agency several years ago. We developed a technology where 5 pathogens can be tested in one test. Okay, with a high resolution melting. We licensed this technology to a Malaysian company, but unfortunately, it was not commercialized because of the COVID. They were all focusing on COVID-19 and testing. Thanks for bringing up this question, Ansela.
And Dr. Rage, due to the time limitation, so now let's just answer the last 3 questions from the Juan. Is
okay. There is one question from Miguel. My questions are, I don't know whether I missed some question. Yeah. Okay, my questions are as follow. Can molecular diagnosis be integrated in veterinary? Yeah, veterinary medicine or aquaculture doesn't matter. I have developed a canine parvovirus. One of my students developed last year, developed a test for canine parvovirus. And aquaculture, I am basically focused on maybe if you have missed the last slide, the multiplex, they are all for WSSV and EHP. And I don't know whether you are from Philippines. If you are from Philippines, I work with the University of Santo Tomas and the National Fishery Development Research Institute. SIFTEC Iloilo, I have a strong collaboration with the Philippines. We are all working on that. Molecular diagnostic cost more or less similar to PCR. Earlier it was expensive, but the cost will come down. because there are so many products in the market, so much competition. CRISPR-based diagnostic, not many tests are on the market right now, but very soon, there are therapeutics already approved by FDA in place. As far as I know, the CRISPR-based diagnostic tests are not still in the market. I wish we will probably available. Maybe I don't know to best of my knowledge, but very soon you will see that. Attendance link. These are all administrative questions. Attendance link.
Yeah, you can just jump to you have 3 questions.
Yeah, in Uganda, the wife of a late Hana Sugani has accused of family tampering. Yeah, I think Samuel, I know the tampering in some places it is possible. I have watched a movie. I just last thing that I want to share. I watched a movie where the detectives are unable to find the culprit, and then they have one suspect who is also part of the investigative team. And they take the blood sample, the DNA does not match with the DNA from the crime scene. But they understand that this guy undergone a stem cell transplant. You know, when you have the stem cell transplant, your blood will be from the donor, not from your own. So when you pick up the hair, because our skin, because they are all not renewed like blood cells, when they pick up the hair, then they confirm that he is actually the perpetrator. So these are all possible in some countries, yeah. But I don't want to comment, yeah. This happened in India also, one very famous case where one of the famous politician, he had a illegitimate son. He sued him. He was denying for a long time. Then the DNA test showed, in fact, he was his father. And finally he married at the age of 80. He married his mother. They became a family. I think he is no more. Yeah, and a lot of stories we can talk about the real things. So I think photo session now, the last comment. Yeah. Thank you guys. I know like you have a lot of questions, but for want of time, I will leave the floor to Miss Luna and her team. Yeah. Thank you very much.
Okay, thank you. Thank you so much for informative answer. So next, let's move to the group photo section. Therefore, this part will be the final part of our webinar today. So everyone, please turn on your camera. We will do the screenshot. Please turn on your camera. And after the group photo section, we will post the attendance link in the chat box. So please turn on your camera. We will finish this group photo section. Yes, please let me see your screen. And
I see only 4 or 5 people.
wait a second. Yes, and Dr. Ray, please close your screen sharing because we will do the screenshot.
Do I need to share close? I think I thought already closed.
Let me check.
Is it me?
Everyone, please turn on your camera so we can see your face and do the screenshot. Okay, please turn on your camera. After the group photo section, we'll post the attendance link in the chat box. So please turn on your camera as soon as possible. We'll do the screenshot. Okay, do you have any participant turn on the camera? Let's give some time to our participant open the camera. Okay, everyone, please turn on your camera. We need to see your face in the screen and do a screenshot group photo.
Are you still sharing your screen, Miss Luna?
Yeah, I stopped to sharing the screen because I need to do the screenshot for the old people.
Oh, okay.
Okay, so yeah, so now I will do the first screenshot. Yeah, I will counting down. Okay, 3,